Hello friend, welcome you all in this session. Myself BP Shah, PGT Biology, Kendriya Vidyalaya Kesapuram. Today I am going to continue my last session lecture, that is respiration in plant, included in chapter 14 of plant physiology, unit 4 in class 11th NCERT. In my previous session, at the end of session, I have asked some questions related to the glycolysis and the main chapter of respiration. I hope today all you will be able to answer these questions. Now, first of all, what are the questions previously asked? My first question was, what are the main steps of aerobic respiration? There are two types of respiration, we have already seen, aerobic and anaerobic, and their steps already discussed. The next question is, what is the net ATP production in glycolysis? The third one question, in which cell organelles does respiration takes place? Where does the fourth question is, where does the aerobic and anaerobic respiration takes place? And the last question was, we commonly call ATP as the energy currency of the cell. Why? Why it is called energy currency? Can you think of the some other energy carrier present in the cell? Give name any two. The answer of these questions are, the main steps of aerobic respiration which takes place in cytoplasm as well as in mitochondria are glycolysis, that is also called EMP pathway, where the one glucose molecule converted into two molecules of pyruvic acid. In presence of oxygen, that pyruvic acid enter into the Krebs cycle. So, the second step is Krebs cycle. The third one step, electron transport system, electron transport chain also called, and the fourth one oxidative phosphorylation. So, this is the answer of my first question. Now, the second question is, what is the net ATP production in glycolysis? I have already discussed the net ATP production with the help of substrate, where the ATP is used or consumed. In entire process of glycolysis, the 2 ATP is consumed and 4 ATP is gained. So, the total plus 4 ATP minus 2 ATP, that is 2 ATP. So, the answer is 2 ATP. Now, the third one question is, where does aerobic and anaerobic respiration takes place? The aerobic respiration takes place in cytoplasm, where glycolysis takes place, and the end product of the glycolysis, it enters into the mitochondria. So, for aerobic respiration, the cell requires cytoplasm and mitochondria. For anaerobic respiration, which is called fermentation, also it takes place in cytoplasm. Now, the fourth question is, we commonly call ATP as the energy currency of the cell. Why? Can you think some other energy carrier present in the cell? Give two example, any two example. We already discussed the formation of ATP, which is made by adenosine and 3 phosphate group, which is made by nucleotide, adenine, sugar, and 3 phosphate groups are there. It is for many steps in the respiration. So, it is called commonly called ATP energy currency of the cell, and this ATP formed by the glycolysis or Krebs cycle or electron transport chain used whenever it needed, just like currency, when we need we expand the currency. So, the ATP acts as a energy currency of the cell, because it helps in the various metabolic process where the ATP is needed. Yes, the second one question answer is yes. Beside this ATP, ADP, adenosine diphosphate, and adenosine monophosphate are the other carrier present in the plant cell, which also act as a energy currency. Now, the actual lesson, today's actual lesson is different types of respiration. That is, there are two types of respiration. They are aerobic respiration and anaerobic respiration. Aerobic respiration, it is already discussed in class, lower classes like 10th. It is a respiration in which glucose molecule broken down in the presence of oxygen to release carbon dioxide, water and energy. It takes place in most of the organism. The reaction is glucose molecule in presence of oxygen, it gives carbon dioxide, water and the energy. This energy in the form of ATP. Now, the second process is called anaerobic respiration. Is the respiration in which glucose molecule is broken down in absence of oxygen. To release, it depends upon the where the anaerobic respiration takes place, release alcohol, carbon dioxide and energy, which is least in comparison to the aerobic respiration. It takes place in yeast. The reaction is glucose molecule in absence of oxygen, it form alcohol, that is ethyl alcohol, and carbon dioxide and energy, that is 2 ATP. As we have already said, this takes place where the reaction is going on. In case of yeast, the product is alcohol, but in case of the muscle cell, at the time of fatigue of muscles, the glucose molecule is not completely broke down. In absence of oxygen, it releases lactic acid and energy. So, glucose molecule in absence of oxygen takes place inside the muscle cell, it forms lactic acid and energy. This energy is also 2 ATP, which is less in comparison to the where the oxygen is used, that is aerobic respiration. Now, the main topic here, that is the anaerobic respiration, which is also called fermentation. In this process, the oxygen is not utilized, even it is present. Here the pyruvic acid, which is the end product of the glycolysis or the EMP pathway, is incompletely oxidized, and the end product differ in the different tissues or the organisms. There are two types of fermentation, that is alcoholic fermentation and lactic acid fermentation. Now, one by one, that is the lactic acid fermentation and alcoholic fermentation, which takes place in the different types of tissues or the organism. First of all, we see the alcoholic fermentation. The alcoholic fermentation as the term indicate, fermentation in absence of oxygen, and the product is C2H5OH, that is ethyl alcohol. it takes place, example of alcoholic takes place many of the microorganisms. Organisms and yeast, that is a fungi. What happened in alcoholic fermentation? The end product of the glycolysis, that is pyruvic acid, we have already discussed their structure, that is CH3 CO COOH, that is pyruvic acid. Two molecules of pyruvic acid is formed in glycolysis. It undergoes decarboxylation. So, the enzyme used, that is called decarboxylase. Here the substrate is pyruvic acid. So, we can say pyruvate decarboxylase. It need activators like Mg++ and TPP. First of all, it converted into acetaldehyde. This acetaldehyde, as the term indicate, the functional group is aldehyde. The structure is CH3 CHO. This is the functional group of aldehyde, and 2 molecules of carbon dioxide is released and 2 molecule of acetaldehyde is formed. So, first reaction is the pyruvic acid converted into acetaldehyde. Then this acetaldehyde again undergoes the another reaction, that is the second reaction is this acetaldehyde, it is also called ethanol for aldehyde. Ethanol, it is going to be reduced, that is the reaction is 2 molecules of acetaldehyde CHO, it combined with 2 NADH2 which is the product of glycolysis. In one step of the glycolysis, the NAD combined with the proton molecule and it form NADH2 H plus H plus. So, this acetaldehyde going to be reduced with the help of this NADH2 in presence of enzyme dehydrogenase. The product is alcohol. So, we can say alcoholic dehydrogenase या alcohol dehydrogenase. Now, it form CH3 CH2 OH plus twice NAD. This 2 hydrogen is used for the conversion of acetaldehyde into the ethyl alcohol, that is called also called ethanol. So, the end product of alcoholic fermentation, that is pyruvic acid, 2 molecule of pyruvic acid, it is converted into 2 molecule of ethyl alcohol and NAD, which is again used for the further reaction. So, we can say finally, in alcoholic fermentation the net reaction will be net reaction of alcoholic fermentation final from the start from the glucose molecule C6H12O6 2 ADP plus 2 pi that convert into CH3 CH2 OH plus 2 CO2 plus 2 ATP plus 2 H2O. So, now we are able to say that in alcoholic fermentation glucose molecule is converted into ethyl alcohol, 2 ATP is formed. When one glucose molecule is going to be degraded. So, that now this is the first step of the anaerobic respiration. The second step, the second product is lactic acid. So, this is called lactic acid fermentation. The lactic acid fermentation, it is formed in muscle tissue, and most commonly used in our day to day life in our household item, that is formation of curd using lactic acid bacteria. It also takes place in some fungi. So, these are the common example where the lactic acid fermentation takes place. Now, what happened in the lactic acid fermentation? First of all, the pyruvate which I have already discussed, CH3 CO COOH, that is the end product of glycolysis. It directly combined with NADH and it directly converted into CH3 CHOH COOH plus NAD plus. Here the these two hydrogen from the NAD H2 is used. So, the reaction is dehydrogenase. The product is lactic acid. So, the enzyme better to say that is lactate dehydrogenase. For this reaction Zn plus plus is required and also FMN is required. So, the in lactic acid formation pyruvic acid converted into lactic acid, 2 molecule of pyruvic acid converted into 2 molecule of lactic acid. So, we can summarize this reaction. So, the net reaction of lactate fermentation, that is starting from the glucose C6H12O6, it is used 2 ADP plus 2 Pi and it converted into CH3 CHOH COOH, that is lactic acid plus 2 ATP and twice H2O. So, the glucose molecule gives 2 molecules of lactic acid and 2 ATP, which I have already asked in my previous question, how much ATP is formed in the anaerobic respiration, that is the 2 ATP is formed in this process. Okay. So, the two type of reaction it is here also, the alcoholic fermentation and lactic fermentation, the product is lactic acid and ethyl alcohol. Now, come to the next reaction, that is for aerobic respiration. Aerobic respiration. In my first slide, we have seen there are two type of respiration, anaerobic and aerobic. This, as the term indicate, the aerobic respiration takes place in presence of oxygen. And the product will be, the glucose molecule is going to be break down. Here the glucose molecule in presence of oxygen completely break down, but in case of anaerobic respiration there is a partial breakdown of the glucose molecule. But here the in aerobic respiration the glucose molecule completely break down in presence of oxygen. Now, it form carbon dioxide, water and huge amount of energy, that is in somewhere 36 and 38 ATP is formed, which I will discuss later on, where the 36 or 38 ATP is formed. So, we can balance it 1 molecule of glucose combined with the 6 molecule of oxygen, it forms 6 molecule of carbon dioxide and 6 molecules of water and 38 ATP is formed in complete breakdown of the glucose molecule. And the pathway of the aerobic respiration, that is common pathway for aerobic and anaerobic, that is called glycolysis. So, for aerobic respiration also started with the end product of the glycolysis, that is pyruvic acid. 2 molecule of pyruvic acid is taken into consideration, which is the end product of the glycolysis. Now, this pyruvic acid enter into the Krebs cycle. This Krebs cycle was discovered by scientist Krebs, as the name indicate, and the name of the cycle is called Krebs cycle. But this Krebs cycle takes place in mitochondria, where all the enzymes of the cycle are present in the mitochondrial matrix, as well as their other steps are present in the mitochondrial molecules. These mitochondria are, we know in the chapter cell structure, mitochondria is the double membrane structure. The outer membrane is smooth and the inner membrane, that is folded. You have already read in chapter cytology in different type of cell organelles. Here only the outline is outer membrane, this one is called inner membrane. the inner membrane are folded. It is also called cristae. In this cristae, there is a pinhead like structure are found. Do you know what is this? Yes, you have already read in the chapter cytology. This is called this is called oxysome. This oxysome are the site of electron transport chain or oxidative phosphorylation. I have already discussed in previous class of the photosynthesis. There are three types of phosphorylation. The meaning of phosphorylation, that is formation of ATP. These ATP are formed at three levels. One is called substrate level phosphorylation. In photosynthesis, we have seen photophosphorylation, that is cyclic and non-cyclic phosphorylation. Here you will see the ATP is formed, but here the oxidative phosphorylation takes place in the oxysome, and the inside the inner membrane there is matrix. In mitochondrial matrix, all the enzymes for the Krebs cycle are present in mitochondrial matrix, and electron transport chain, which is takes place with the help of cytochrome and various types of iron, that is the ferric and ferrous, which is found in the oxysome. The oxysome structure of this type this one is the head, it is the stalk, it is the base. So, all the enzymes are present here at the head of the regions. This is the structure of oxysome. Here is the outer side of the mitochondria, that is called outer membrane. Here is the proton channels, this one is the proton channels, and the oxysome are made by two particles, that is F0 and F1 particles. This one is the head. This head contain ATPase enzyme. This ATPase enzyme help in the synthesis of ATP, which combine ADP and PI and form ATP. This one is the stalk, that is made by F5 and F6, and the base one is called F0. This one is the inner mitochondrial matrix membrane, here is the matrix. So, the all enzymes are present for the Krebs cycle, which is we will discuss. The matrix contain all the enzymes for the Krebs cycle, and the oxidative phosphorylation or the electron transport chain are present in matrix as well as in the oxysome. Now, what is the fate of pyruvic acid in aerobic respiration? Before participating in Krebs cycle, the pyruvic acid enter into the mitochondria not directly. This pyruvic acid undergoes decarboxylation. Means from the pyruvic acid carbon dioxide is released. Pyruvic acid, which is 3 carbon compound, when one carbon is released in the form of carbon dioxide, it forms 2 carbon compound, that is called acetyl coenzyme. Acetyl coenzyme A. So, before going to be participation in Krebs cycle, the pyruvic acid undergo decarboxylation, and for 2 molecule of acetyl coenzyme, this acetyl coenzyme now able to enter into the mitochondria, that is mitochondrial matrix, where the further reaction of Krebs cycle takes place. So, here you think the pyruvic acid, which is the product of glycolysis, not directly enter into the mitochondria. Before the participation in Krebs cycle, pyruvic acid first of all undergoes decarboxylation process, and in that cases the combination of acetyl coenzyme, and conversion of pyruvic acid into the acetyl coenzyme takes place. Here the reaction of the decarboxylation process, that is, it is pyruvic acid COOH, it form acetyl coenzyme CH3 COSCOA, that is called acetyl coenzyme A, and here there is the release of proton in the form of hydrogen, which one is accepted by NAD and converted into NADH plus H plus, and this carbon dioxide of the pyruvic acid also removed. So, here the two reaction is going on. First of all, removal of carbon dioxide. So, this process is called decarboxylation. Removal of proton, that is hydrogen, which is accepted by NAD. So, this process is called reduction. So, carboxylation and reduction process takes place. So, the enzyme used for this, that is called dehydrogenase complex. Dehydrogenase complex. This complex term used for both cases, release of hydrogen as well as carbon dioxide. This carbon dioxide, and there is the use of coenzyme HSCOA. This hydrogen and this hydrogen here used H, this 2 H, and the carbon dioxide is going to be released. So, the product is the pyruvic acid now converted into acetyl coenzyme, 2 molecules of acetyl coenzyme. This acetyl coenzyme now able to enter into the mitochondria, and this NAD and NADH2 is used in oxidative phosphorylation. And you know that, which I have already discussed in my last class, this NADH2 after the oxidative phosphorylation it gives 3 ATP. One molecule gave 3 ATP, here the 2 molecule of NADH2 is formed. So, the net gain will be, that is the 6 ATP. So, we can say now here in glycolysis there is the formation of 2 ATP and 2 molecule of NADH2. Here also the 2 molecule of NADH2 is formed, which will be find in my release of energy chapter at the end of this. That is, in this process the 2 molecule of NADH2 is formed. So, total NADH2 molecule in glycolysis and oxidative decarboxylation, 2 molecule at the glycolysis and 2 molecule at oxidative carboxylation. So, total 4 NADH2 is gained at the end of the oxidative decarboxylation. Now, when the pyruvic acid enter into the mitochondria with the help of acetyl coenzyme, which is the product. Now, the cycle is going on, that cycle was discovered by Krebs. So, this is called Krebs cycle. In this cycle, the product which is formed firstly, which contain 3 acid molecule. So, this cycle 3 acid molecules are there. So, this cycle is also called tricarboxylic acid cycle. The product is called citric acid, product name is citric acid. So, this cycle is also called citric acid cycle. So, the Krebs cycle also named as tricarboxylic acid cycle, citric acid cycle and Krebs cycle. Here the acetyl coenzyme which enter into the Krebs cycle, 2 reaction takes place. Firstly, the Krebs cycle and second one step, that is called terminal oxidation. Terminal oxidation, because as the term indicate it is terminal, it takes place at the end of the respiration. So, it is called terminal oxidation and oxidation takes place. This terminal oxidation also divided into 2 parts. The one part is called electron transport chain or electron transport system. Electron transport system, and the another process which is sequentially attached with the electron transport chain, that is called oxidative phosphorylation. So, this electron transport chain and the oxidative phosphorylation, it takes place, the oxidative phosphorylation takes place in the oxygen, and inside the matrix all the enzymes for the electron transport chains and other cytochromes are found here in the mitochondria. So, the next will be the pyruvic acid in the form of acetyl coenzyme, which is the product, it is going on into the Krebs cycle. The Krebs cycle, it is the common, now come to the Krebs cycle. Here it consists of 2 distinct mutually dependent process, that is called Krebs cycle and the terminal oxidation. which I have already discussed. We have discussed it. Before participating tricarboxylic cycle, pyruvic acid enter into the mitochondria and decarboxylation process takes place with the combination of one molecules of coenzyme, which already discussed. Pyruvic acid, the first acid first undergoes oxidative decarboxylation to form acyl coenzyme. During this process, NAD is reduced, NADH, NH plus and CO2 is released. The acyl coenzyme enters into the Krebs cycle for further degradation. Now, steps of the Krebs cycle in eukaryote, it takes place, it operates in the matrix of the mitochondria. The one mass question is, what is the substrate of Krebs cycle? That is acyl coenzyme, because the end product of the pyruvic acid of the end product of the glycolysis, which are not able to enter into the mitochondria, it first of all converted into acyl coenzyme and then converted into acyl coenzyme that will be entered into the Krebs cycle. The Krebs cycle was discovered in 1949 by Sir Hans Krebs. It was firstly seen by Krebs in pigeon muscles. For this, Krebs awarded by Nobel Prize in 1953. It is called, as we have discussed, TCA cycle, tricarboxylic acid cycle. It is also called citric acid cycle. Now, the most important thing for the Krebs cycle, this cycle, it is a common oxidative pathway for carbohydrate, protein and fat, because in glycolysis the pyruvic acid is formed and finally, we have seen my respiration lesson first, the carbohydrate, protein as well as fat all takes part in the Krebs cycle. So, we can say it is the common pathway for the carbohydrate, protein and fat. In this Krebs cycle, some intermediate product is formed, which is synthesized for different essential material of the biomolecules like glutamate, aspartate. This product are formed with the help of the intermediate product of the Krebs cycle, that is alpha ketogluteric acid. It is discussed in the chapter mineral nutrition. When the biological nitrogen fixation takes place, the product will be the ammonia. The N2 is converted into ammonia. This process is called ammonification. You know that ammonia is highly toxic plant as well as human being. This ammonia is converted into nitrite and nitrate. These are the usable form for the plant. This process is called nitrification. Sorry. Now, when the ammonia is converted into nitrite and nitrate, then no problem, plant are easily absorbed, readily absorb this and make various types of biological substance. When the ammonia concentration is going to be high in case of the plant, then this ammonia combined with alpha ketogluteric acid, which is the one of the intermediate product of the Krebs cycle, that converted into glutamic acid. And the reductive amination and transamination product glutamic acid as well as aspartic acid is formed, which is the stable product and useful for the plant as well as human being. So, the various intermediate product of the Krebs cycle are used to synthesize important biomolecules in the living system. Now, the Krebs cycle, which have various types of steps in eukaryote, in mitochondrial matrix, Krebs cycle takes place, and there are 9 successive steps involved in the Krebs cycle, which start from the acyl coenzyme, no doubt. Now, the steps are which involved in the Krebs cycle, that is, first step is condensation. In condensation, what happened? The acyl coenzyme, which is the product, it combine with oxaloacetic acid and this oxaloacetic acid, it form citric acid. So, the first reaction is called condensation, which will be discussed later on. So, in condensation the two molecules is going to be combined, that is oxaloacetic acid plus acyl coenzyme. It form citric acid, which have 3 COH group. So, this cycle is called tricarboxylic acid cycle. Now, the second step in the Krebs cycle, that is isomerization. This citric acid is finally form isocitric acid with the help of enzyme aconitase, and the intermediate product is called cis aconitic acid, and this citric acid is converted into isocitric acid. So, with the help of isomerization, in this case we will see in my next class, here water is going to be added or removed, and when it is going to be removed from the citric acid, it forms cis aconitic acid. When it is going to be added, it forms isocitric acid. So, the second steps of the Krebs cycle is isomerization. Now, the third step is dehydrogenation. As the term indicate, here the removal of hydrogen, which will be accepted by NAD. The third one step is dehydrogenation, and the fourth one step is decarboxylation. As the term indicate, in decarboxylation process, carbon dioxide is going to be removed, and the step is called decarboxylation. So, in 9 steps, these are the 4 steps, and the 5th step is oxidative decarboxylation. Oxidative decarboxylation takes place. In oxidative decarboxylation, as the term indicate, there is the removal of carbon dioxide, and there the oxidation takes place. Now, 6th one step is very important. Here the substrate level ATP synthesis takes place. Here the formation of JTP, which finally converted into ATP. So, the 6th reaction is substrate level ATP GTP synthesis. Here the GDP guanosine diphosphate, it combined with PI. Firstly, it formed GTP guanosine triphosphate. This GTP again converted into ATP. So, here ATP is formed. So, we first of all say that where there is the formation of ATP, that process is called phosphorylation. Here substrate form the ATP. So, this one is called substrate level formation of ATP. 2 GTP is formed, which is finally converted into 2 ATP. Now, the 7th process in case of Krebs cycle, that is dehydrogenation. As the term indicate, in dehydrogenation process, the hydrogen molecules is released. In this process, the dehydrogenation, NADP, NAD is not used. Here FAD is used to accept this proton, and it converted into FADH2. These are the reduced coenzyme FADH2. It converted into 2 ATP in oxidative decarboxylation process. So, it is the first step where for the acceptance of hydrogen atom, NAD is not used, FAD is used and converted into ATP. Now, the 8th one reaction is hydration. As the term indicate, hydrogen is going to be added, and the last one reaction of the Krebs cycle, that is dehydrogenation. Where hydrogen molecule is released, which is one is accepted by NAD and converted into NADH plus H plus. So, these are the 9 steps which we will discuss in our next classes. Now, think over these questions. What are the differences between glycolysis and fermentation? Think over it. Now, you will be able to answer this question, what is glycolysis, what is fermentation. What are the differences between aerobic respiration and fermentation? The third one is aerobic respiration and anaerobic respiration. My next question is, what are the end product of fermentation in case of yeast? And the last question in this session, that is, name 2 instances where lactic acid is formed by the fermentation. The one instance you already know in our home, when the milk is converted into curd. Now, I hope you will be able to answer these questions in my next session. Thank you.